Hello, and a very warm welcome to everyone joining us for this Select Science webinar titled Creating Breakthrough Treatments with siRNA, Learner Biopharma's Liver Regeneration Story. My name is Cameron Smith-Craig. I'll be moderating today's presentation, and I'm delighted to be joined by our guest speakers, Dr. Torsten Wusterfeld and Dr. Si Tan from Learner Biopharma. Please feel free to submit any questions you may have during the webinar. To do this, simply hover over the speech bubble icon to the left of your screen and click to open the submission tool. During the webinar, we will also be posting a few poll questions. If you could take 2 minutes to submit your answer when prompted, it would be greatly appreciated. Thank you. So without further delay, I'm delighted to hand over to Dr. Wusterfeld and Dr. Tan, and would like to thank them both for presenting to us today.
Thank you, Cameron, for the introduction. I'm Si Hui. I'm the VP of Biology at Learner Biopharma. And today, Torsten and I, Torsten is the co-founder advisor of the company, and we will be presenting to you how Learner Bio translated novel technologies using this very promising modality called siRNA into a liver regeneration therapeutic. So the first question many of you may ask is, who is Learner Biopharma? We are drug developers of first-in-class RNA therapeutics, and our immediate goal is to treat liver diseases. And we are combining two very promising technology over here. So at the start, with the novel biology, Torsten has come up with this screening platform in mice livers to discover these drug targets that are immediately targetable by siRNA. And we have also built our proprietary siRNA delivery and modification platforms to create a drug that can be given to the patients. And when you combine these two very powerful platforms together, we can generate these first-in-class drugs. And we are very, very honored to be recognized by Clarivate globally as one of the 7 RNA technology companies that have been pushing the frontiers in such innovations. So I think a lot of you have seen these 3 points, and hopefully this will be some pointers and some learnings that you can take away from us. Through our sharing of our story and our journey, we hope to help you see how siRNA is a very powerful modality, and it really has immense potential in going after a lot of previously undruggable targets that have been uncovered in biology. And we're also sharing how we generate these first-in-class therapeutics through our in-house thought processes, platforms, and cascades. And we look forward to the Q&A and happy to share with you more at the later session. And with this, I'll turn over the mic to Torsten, who will talk about the liver disease and platform. Torsten, please.
Thank you so much, Si Hui. So first of all, you might be wondering why it's important to target liver disease. It's interesting to see that liver disease, compared to a lot of other major causes of death, is still on the increase. This is here an example from the British Liver Trust. And this is mainly caused by the current obesity epidemic. So we have a lot of patients which are now having the manifestation of the metabolic syndrome in the liver, which is called simply fatty liver disease or non-alcoholic fatty liver disease. And despite we have currently hepatitis B virus vaccination, we have hepatitis C virus drugs, which are very efficient, the amount of people suffering from chronic liver disease is still increasing based on this fatty liver disease. If you think about it globally, cirrhosis, which is kind of nearly at the end stage of the liver disease, we have 120 million people are suffering from this. And we have around 1.3 million deaths per year attributed to chronic liver disease. If you think about liver biology, how it happens, how from a healthy liver we come to this end stage liver disease, we start basically with a healthy liver, but we have under normal disease condition is a chronic damage. So liver cells are dying based on a damage, be it now overnutrition in the obesity case, or hepatitis virus infections, or alcohol. And this leads to compensatory liver regeneration. So you might have heard already by the Prometheus Greek mythology that the liver has a very nice regenerative capacity. But nevertheless, over the time, we see like an accumulation of fibrotic tissue, which kind of like substitutes for the death of like hepatocytes. And if this continues over a long time, we see like the development of liver cirrhosis. And this is like the point where we go into end-stage liver disease. And one outcome of this can be the liver failure or liver cancer, which is an end stage. The only curative treatment which we have for all of like this like chronic liver disease, end-stage liver disease so far is liver transplantation. But I have to mention that there are not for sure enough donor organs for the demand which we have. On the other hand, often liver patients with end-stage liver disease, they're very fragile and they're not really up to major surgery, which is an additional contribution that like at this stage the outcomes are very very dark. If you think about like what the physician classically would do is like that at the stage of liver fibrosis, we would treat kind of like the etiology. So in the case of like if you overeat, there would be like the treatment of like, okay, reduce your food income, make more sport. If you drink too much alcohol, reduce alcohol intake in this. And at that stage, like if you would follow this advices, you can have like regression from the liver fibrotic areas back to more like a healthy liver. Other point is like liver cirrhosis, you have more like a stronger exhaustion of this like capacity. So one thing is like to improve liver functions. People try different approaches in this direction. And one would be like to enhance the liver regeneration, to come back and counteract like this chronic liver disease. As if I mentioned, like chronic liver disease progression can be very slow. So it can be decades because the liver has such good intrinsic regenerative capacity. So if we think about the liver, as I mentioned, it has normally an amazing intrinsic regeneration capacity. This is most and best shown by a classical partial hepatectomy experiment, which was established in 1931 by Anders and Higgins. And what they did is like they surgically removed 2/3 of the liver from a rat, and they saw like that the rest of the liver can kind of like regrow, and the full function of the liver can be reinstated. We basically use this even in our days, this advantage of this regenerative capacity of the liver, if you think about like living donor transplantations. So like if a donor gives a part of the liver, for example, a relative, on one hand, his liver gets surgically a part removed, but it regenerates. And even if this other part is smaller than the recipient need, it can also like to a certain point like regrow and regenerate. So we already utilizing liver regeneration in this point as a treatment modality. The sad thing to say is that like liver regeneration, this capacity seems to be degrading over time. So during aging, we see like that the older we get, the regeneration takes longer of the liver. So there's a decline in this. And chronic liver disease seems to, in a simple word to say, like it's an acceleration of this process. Like we see also like a faster decline of this. The idea is like that you have an accumulation of things which inhibit the correct or like the original regenerative capacity. And the idea is to target these inhibitors. So this we go from a low proliferation rate to a high proliferation rate, and liver regeneration can compensate against any damage. And one approach to do this very efficiently is to use GalNAc siRNAs. So we will a little bit tell you more about this GalNAc siRNAs, but what they allow is like a specific delivery to hepatocytes of this siRNA. So if we have knockdown of our target, we can then enhance regeneration of this hepatocytes. And we can show you like how we think to identify such targets to enhance this regeneration capacity, to kind of like return the clock a little bit of this aging or accelerated aging by chronic liver disease. So what we do is like we do in vivo functional screening. The classical idea is a lot of people go this, that you have a pathway in mind and you follow up the pathway to understand the biology of the liver and liver biology, and then maybe utilize this for liver regeneration. But this approach also it gave us a good understanding of the regenerative process. So far, didn't really lead to developments for the clinic. So another idea is like to let the biology tell us what might be good targets for such purpose. So in this case, you do an unbiased screen. What we do, we deliver a pool of shRNAs to the liver so that we have stable integration in a subset of hepatocytes. We then do like chronic damage, and any form of chronic damage can be used. For example, we can put mice on high-fat diet, or we can also use like chemical compounds or alcohol to induce chronic liver disease. And then after time, we harvest the liver. And the idea is that like if the knockdown by a certain shRNA enhances the regenerative capacity of this hepatocytes, gives them an advantage in this like kind of like negative environment of chronic damage, then we will see an enrichment of this shRNA compared to the starting situation. And harvesting the liver, isolating the DNA, amplifying this like expression cassettes for the shRNA, we can compare this to the starting point and can, through sequencing, identify shRNAs which are enriched in this point. And the good thing is like this is an shRNA, and shRNA is a precursor of siRNA. There is a very fast translation to siRNA therapeutics. So if you have a target, we don't have to go the way to identify, for example, small molecule inhibitors or even like antibodies or whatever to try to inhibit the target. No, we can directly use siRNAs to have the same effect which we saw from the shRNAs. Now I will now go like to show some examples of this for this target selection validation. So first of all, what we do is like if we have a target out of this shRNA, so we have an enrichment for this shRNA, we look into the literature if there's any link, for example, in regards to cancer. Anything which enhances proliferation has always like, in theory, could have an impact also like on being like more like a pro-cancerogenic entity. In this case, what we prefer to see is like that patients who have like a low expression of the target have a better survival outcome, and that there's no direct connection, that this is like inducing like transformation of cells. The other thing is like the shRNAs which coming from the screen have to be validated that they have efficient knockdown on the target. It's like there is off-target effects of shRNA, so they can have this. We want to be sure that we have an on-target effect and that we have at least 2 independent shRNAs for the same target, which guarantees that there's no off-target effects. We then established like the combination of in vitro as well as in vivo validation experiments. On one hand, in vitro, we take cell lines of immortalized hepatocytes, and we can use mouse hepatocytes, but also human hepatocytes, and we can look if the knockdown of the target enhances proliferation of these hepatocytes in vitro. We have EDO incorporation assays, doubling time we can analyze, we can use wound healing assay, and we also can look at the transcriptomic, what is the effect on this. And as we can do this in mouse and in human, we can only see if there's translatability from mouse to the human system. We then, if there's a good validation in vitro, move on and then go back to the in vivo mouse system. And in this case, we take advantage of a FRH knockout mouse. I don't want to go here in details. The advantage of the FRH knockout mouse is like that we can repopulate the mouse liver so that every hepatocyte expresses a shRNA of our interest. So we have the target, we have the shRNA against this target, we repopulate this mouse completely, and then we can look if we have classical partial hepatome experiment, do we see faster regeneration, regrowth of the liver? We can also see under different chronic liver damaging conditions, do we see that there's like reduced fibrosis development, and even like this repopulation in itself, we can compare to control shRNA if the repopulation happens faster. And this gives us like a good understanding if this target has like therapeutic value. The other thing is like coming back to the safety issue, which I mentioned before, we can let mice sit with this knockdown for long term and see, do this liver architecture is like affected? Does it lead on long term to tumor development? Is liver size in relation to the body size in the correct ratio? And if all this is good, we have like a very good safety for this target and can continue. I give you one now example in a chronic damaging condition experiment. So one of our targets, we repopulate this mouse livers, we expose them, this mice, to chronic chemical damage by giving thioacetamide. And what we see is like that we have a strong reduction in the fibrosis level. And what we also found for this for our target is like that the same thing we see if instead of like a chemical damage, we use Western diet, a high-fat, high-fructose diet, we see also reduction of fibrosis development. And if we do partial hepatomy, we see like faster regeneration after the surgical removal. So this target has a very, very strong pro-regenerative effect. And as we said, like in the vitro validation, as we look like in human and mouse hepatocytes, we know that this effect is conserved between the species. Okay. So this comes to this kind of like platform technology. So the screening setups, we can do like small focused screenings, but what we also did is like genome-wide screening for targets, like a library which is covering the whole genome, and we can also set it up in an inducible system, in an intervention setup. So like the siRNA expression only goes on after we have a disease stage. And we can any form of like disease modeling, chronic liver disease modeling system use, be it like a Western diet model, be it like a alcohol-induced system or chemical-induced chronic damage. And through this, we managed to identify a large number of high potential targets, which then can be translated into siRNA therapies. And our top target so already is translated into a preclinical siRNA, which specifically can be delivered to hepatocytes. I think I now hand over to Sihui. Thank you.
Thank you, Thorsten, for the very nice overview of the liver biology and our target discovery platform. So I'll take over here and give a brief introduction about, what is siRNA and what is the history behind it. So this is a slide I borrowed from my chemistry colleagues who knows way more about siRNA than I do. Now I'll try and do this great history justice. So the first siRNA construct was actually discovered not too long ago in the late 90s. And very quickly, you can see that Craig Mello and Andy Fire actually won the Nobel Prize for this because people realized the potential of this siRNA mechanism. And along the way, people also cracked at the puzzle of how to deliver these siRNAs, because these siRNAs are actually double-stranded molecules, as you can see in this picture here, that would not go through the cell membrane. So how do you deliver it into the cell in order for it to exert its effect? And here, the delivery mystery was solved with LNP as well as this ligand called GalNAc. So GalNAc is a sugar moiety that could be taken up by a receptor expressed on the hepatocytes. So this receptor is also exclusively expressed in hepatocytes throughout the body. So you can imagine that this confers huge specificity for the liver hepatocytes for our siRNA drugs. And with this part of the puzzle cracked, along with other delivery modalities, next came the advancement in chemical modifications. So why do you need to modify siRNA? Our high school biology would tell us that once you put RNA into your blood, unmodified RNA into your blood, it gets broken down immediately, almost immediately by nucleases. So these chemical modifications serve to make these molecules more durable so that they can last longer in the body. And the other important thing is these modifications actually remove the immunogenicity effect. So imagine this RNA interference mechanism was here to protect us from bacterial and viral infections. And these chemical modifications would help the double-stranded siRNA evade this immunogenic response. So this is also a huge progress towards generating the siRNA therapeutic. So currently, you can see now we're in the 2000s, and since 2018, there's been a flurry of siRNA products, and towards the later stage, more of GalNAc siRNA products that have received FDA approval and gone to serve the patients. So at Learner Bio, our formula for doing first-in-class siRNA drug development is to go after novel targets that Torsten's platform has nicely developed and identified, and use our siRNA chemistry platform to generate quality molecules with standing on the shoulders of all these giants that have come before us. So with this, we are using innovative solutions to tackle the large issue of liver diseases. And I will also say that with Torsten's screening platform, it was really nice to translate into siRNA because you could appreciate how closely shRNA and siRNA are linked. And this is really kudos to the design for designing with the end in mind. So I told you our formula, but exactly how did we do it? What's the method? We built out a set of technology platforms to drive our drug development. So the cause identification platform was what you just saw that happened in the mouse. And then we have our OSCAR and our SMOP. These help us to design the siRNA sequence itself, as well as the chemical modifications that decorate this siRNA throughout this siRNA. We also have a ligand discovery platform where we developed our own GalNAc ligand so that we could connect to the siRNA to deliver to the liver. And this is how we operationalize it, so as to say. So here is the journey we've taken to develop our preclinical candidate called LR1. So we've gone through here where we saw how we did the target screen and validation that led to the target nomination. And then moving on, we developed our own GalNAc ligand, and we also developed the siRNA molecule itself. So with the ligand I just mentioned, we use the Navigate platform, and for the siRNA molecule, we designed the sequence with OSCAR and the chemical modifications with SMOP. And this constitutes the hit to lead stage of the drug development process. And once we identified and selected our lead, we brought it into the in vivo preclinical models to look at the therapeutic efficacy of the molecule as well as the target. And when we got positive results that prompted us to move forward, we moved into the IND enabling phase where you do these various set of studies in order to ensure that we have a good drug profile of our molecule. And this process is called the lead to preclinical stage. And once the molecule passes all these tests, we are ready to file IND, which stands for Investigational New Drug. And this then becomes our clinical candidate. And so this entire process constitutes the siRNA drug development. And I'll go through each of them in detail. So first, our GalNAc. This is the enabler of our liver delivery and therefore therapeutics. Like I mentioned, we developed our own proprietary GalNAc, and we compared it against the industry gold standard, which is the triantennary GalNAc. So the target over here is PCSK9. So both GalNAcs carry the same siRNA sequence, and they are all tested in the mice. So with a single injection, you can see that the red and the green lines, they trend very closely together, indicating that they achieve similar levels of internalization and therefore knockdown, and they are also retained for a similar duration as well. So this assures us that we are confident our GalNAc is functionally equivalent to the industry gold standard. And of course, when you develop a drug, like what Torsten showed you, we put a lot of emphasis on the safety profile. So we did a series of safety tests. Here we are showing some mice who have been dosed with very high dosage of 100 milligram per kilogram of the GalNAc siRNA and repeatedly dosed for 5 weeks. So we have the ALT, which is a liver toxicity serum biomarker. And what we want to see is that the levels stay low throughout. And this is indeed what we saw. So our GalNAc is also not toxic in vivo models. And we also did a series of other tests in the human systems, and they all look very safe. And of course, this is our positive control, as we always like to have in our experiment. So now that we have settled the ligand to bring the siRNA into the cells, we need to develop our own siRNA against our novel target. So this is based on using our platforms, the Oscar and the SMART platforms. And because these have been established and running very well at that point in time, we only took 4 months from target nomination to finding our lead. So here are some of the target profiles. siRNA, as we all know, can be very potent and is very potent. So we have our lead asset LR1 here with a picomolar with a 1.1 picomolar IC50. We also had a second lead, which fed a bit worse, but actually it is still very, very potent at almost 20 picomolar. So the other strength and advantage of siRNA is the rationality in not just designing the sequence against your target, but also the very nice off-target profile that you can get just by doing something very routine in lab called RNA-seq. So with the transfection into the cell lines, you can actually see what other genes are also being downregulated other than your target. So before we reached our optimized lead, we had a so-called parent, a precursor. You can see that there was a whole bunch of off targets that fell below the threshold. But after iteration with the SMART platform, where we did some chemical tricks to reduce the off targets, where we repeated the same experiment, you can see that there were just 2 off targets. So we really cleaned up this off target profile. And I also say that our screen happened very quickly because we did all our chemical synthesis, design, etc., all in-house. And our chemistry team really liked the Mermaid 48 here because we could control the timeline and we could generate these oligos at a very efficient manner. So that really aided in the efficiency of the screen. So like I mentioned, after we get our lead molecule, we will do a variety of preclinical tests. So here are some snapshot of what of the data we got. So looking at just the gene knockdown, this actually showed that this is a mouse model of liver failure. So you can see that in the control mice, they actually died within 40 days of liver failure induction. But when we knocked down our target, these mice in the treatment group, they survived for up to a year when we ended the experiment. So there's a very strong protection of against liver failure and ensuring that these mice live when we knock down the target. And if we go look at the Cancer Genome Atlas database of liver cancer patients, we saw that the patients that express our target at a lower level had better prognosis than those who express the target at a higher level. So at a minimum, it assures us of the safety that we would not be aggravating cancer when we knock down our target. Now, on the other hand, it does seem to have a correlation with better mortality benefits. Okay. And we also did a severe liver injury mouse model. And what we could see was that while the control group in black, the weights reduced very quickly and continued decreasing for a week before stabilizing. Those given our GalNAc siRNA in red, they actually had the weight stabilization happen just 3 days after the surgery. And it continued at a higher level throughout the observation period. These mice that were in the treatment group in red also had lower levels of a variety of liver injury biomarkers, such as total bilirubin and ALT. So in this model, we also see a liver protection effect as well as an injury protection benefit. And not to be naggy, but here we are with some safety data again. We have given our drug repeatedly to mice for up to 12 months, and we wanted to ask if there was any difference between the treatment group and the non-treatment group, because we didn't, like Thorsten said, we want to make sure that we don't see neoplastic changes or even just hepatomegaly. So we looked at the gross morphology as well as the as well as the histology, so more microscopic. And we see that the liver sizes between the two groups were virtually identical, and the livers just by looks identical. And so do the H&E tissue sections as well. So our pathologist assured us that there was no difference that he could see. We also had another experiment where we injected hepatocytes with stable target knockdown and transplanted them into immunocompromised mice, which is a very classical tumorigenesis experiment. And we're happy to say that we did not observe any tumor growth when our target was knocked down. We also have other safety experiments as well, but overall, they all tell us that knocking down our target with our asset, LR1, is safe. And we have spoken a lot about how potent the siRNA is. So we've seen this in the human cell line where we have very low IC50s. And in the mice, when we did a dose ranging curve, we had an ED50 of less than 1 milligram per kilogram. So ED50 is the level, the dosage at which you can achieve 50% knockdown of your target. And less than 1 milligram per kilogram is very low by siRNA standards. We've also put these to test in rats and non-human primates and saw a similar dose response as well as potent inhibition at the highest dose of 6 milligram per kilogram. We have moved into preclinical studies, and our rats and NHP data tell us that this siRNA is also very safe, and we have a wide safety window based on the results. So this makes us very comfortable moving forward to do the IND application and etc. Okay, so in just the part that I finished, I've shown you how we've gone from GalNAc and siRNA molecule development and brought it through in vivo preclinical models and done IND enabling. So we are at the tail end of our IND enabling, but so far we have sufficient data to assure us that we have no hurdle. We see no hurdle moving to the final stage of getting the IND and our clinical candidates. So the work does not stop here. We have just presented our lead asset, but actually, I'm sure you appreciate the huge amount of work that's gone into the platform. And we also just mentioned that you could customize the siRNA library, and you could customize the injury model in this Quartz platform. So there is another iteration of the Quartz platform where we looked at the entire genome of targets and put these mice through a NASH modeling, because we know that there's a huge NASH epidemic that's already happened or is going to happen. And we really need some novel ideas about how to address this issue and treat the patients. So with this Quartz platform, we have some results, and when we do the clustering of the hits, you can see that there are some huge biological processes that pop out that are immediately relatable to NASH. So the three main categories would be metabolic processes, and NASH is a metabolic disease. And you have the cell growth and development. Of course, this goes back to the premise of the platform, which is through cell proliferation and regeneration. And there's also a cluster of cytoskeletal and cellular transport. And this actually relates to a feature in NASH called hepatocyte ballooning, where the hepatocyte structures change because of the intensive damage that they have received. And of course, this is mouse data. And when you make drugs in humans, you always want to de-risk by cross-comparing with patients' omics data, which we are doing right now, and putting them through a validation cascade just to get this kind of empirical support and confidence of target of drug development of the target. And of course, what this generates for us is a NASH target pipeline. Okay. And that is the end of our presentation for our story. And just to recap, we are developing first-in-class RNA therapeutics for liver diseases, and we are combining in vivo biology screen that is really letting biology tell the story and telling us what are the actionable drug targets that we can go after with siRNA. And of course, our modality, which we think is very powerful because it can really go after any gene. There is no such thing as undruggable because of this modality. And our overall mission and motivation comes from delivering these innovation-driven treatments that are different from what people have done conventionally, in the hope of serving all these unmet medical needs by the patients. And with that, these are the context of me, Torsten, and our CEO, Kathy. And feel free to reach out if you have any questions or opportunities for collaboration in the future. Thank you.
Okay, thank you both for that interesting presentation. I hope that everyone listening enjoyed the webinar and found it informative. We will now be moving on to the last part of today's webinar, the question and answer session. Just as a reminder, please feel free to continue asking questions using the tab at the left of your screen. Simply hover over that speech bubble icon and click to ask your question. So the first audience question I have for our speakers today is, can you tell us more about the design of your siRNAs? For example, the type of modifications that you have used. Thank you.
So I think I'll take that question. Okay. So with regards to the design of the siRNA, as you know, is a pretty young modality compared to every other modalities, or most of the modalities out there, but it has so much promise. A lot of validation has gone into it. So several of the organic siRNA molecules have actually entered clinical trials and has been validated. So in the first iteration of our chemistry platform, we're using the well-validated modifications of 2'-O-methyl and 2'-fluoro to build this molecule against a novel target. And of course, we are in the works of developing our chemistry platform further to generate more exciting and cutting-edge modifications.
Brilliant, thank you, Suhoi. Torsten, anything to add? If not, I will move on to the next question. Okay, the next submission we have asks, is it possible that there are more than one inhibitor that can reduce liver reproduction? And if so, is one siRNA not enough?
Yeah, I think I can take this question. So in theory, it's possible, for example, that there are several breakthroughs working together. The point I want to mention or to remind, the screening platform is set up like this, that we see enrichment of an shRNA. So in the context of this chronic liver damage, this shRNA has a positive effect on the hepatocyte to expand. So this only shows that even like under these conditions, there will be a positive effect. In the theoretic background of it is like that we hope for sure to hit the kind of like master regulator, so that this master regulator influences the whole kind of like pathway level, and therefore has a very strong effect. And regarding the translatability into humans, as I mentioned, one thing is like to with our in vitro validation system that we see like, okay, mouse hepatocytes getting show faster proliferation, that this is also conserved in the human hepatocytes. And beyond that, for sure, we can also go into liver tissue slides to see if there's iron is applied there, if there's a positive effect. And you can also utilize maybe humanized mice. So like there's a clear setup that we're only following kind of like things which are conserved between mouse and human.
Right. Okay, thank you. I've had another question come in asking, when you move to clinical trials, what will be the sort of dosage regimen that you expect?
Yeah, so the nice thing about siRNA modality is that they operate within very similar constraints and designs. So we don't expect that our GalNAc siRNA would be dosed very differently from the ones that have been approved out there. So we're looking at a dosing frequency of several months, maybe 3 to 6 months for each dose. And of course, the dosage itself, we will wait for the clinical trial results to nail down the exact dosage.
Right, thank you. Taking sort of a step back, more broader here, asking, in your opinion, what are some of the key challenges that haven't yet been fully resolved in the siRNA therapeutic field?
Torsten, I'll take that first.
Thank you, Torsten.
Yes. So, I mean, honestly, the main problem of this field this is linked to as well antisense oligos, guide RNAs for CRISPR, and this is a specific delivery to certain cell type, to certain tissue. So we are in the lucky and privileged situation that everything goes to the liver, and that already with GalNAc, we have a very specific modality how to deliver it specifically to the hepatocyte. So for us, it's not such a concern, but in general, if you want to expand this to other tissue types, other cell types, so like you want to increase regeneration of pancreas, for example, for against diabetes, this is still like a big concern how to make this specific. There are different approaches with lipid nanoparticles to try them to find specific receptors, like how we found on the hepatocytes, but it's still very much a work in progress. Classically, everybody goes for liver. The other thing is like which you can easily deliver is the eye, because you can inject, and it's like immune privileged organ, and the skin, because you can put kind of like a cream situation or something on it. Other organs are a little bit more complex. This is still moving situation there.
Thank you, Dawson. Suhoi, would you like to add?
Yeah, I'll chime in. So I think siRNA therapeutic, the modality is awesome. Like we've said, we're totally in love with it. The one type of disease that is not so suitable for is actually acute diseases, because it does take quite a few days for the knockdown to happen and exert its biological effect. So that actually kind of constrains the kind of diseases we can use siRNA therapeutics for. And of course, in relation to Torsten's earlier point, right, delivery, the other thing about siRNA therapeutics is we have, I think there's so much potential to go after novel biology, like what we have done, but there's so much more novel biology out there. So the field is probably going to branch out into another era where we can really now start testing many decades of biological work coupled with extrahepatic delivery methods to really harness the power of all the investment we've globally put in for understanding how our body works.
Thank you both. I have a follow-up question, sort of leaning on Suhoi, your last point. Could you elaborate on what you consider as novel biology? Thank you.
Novel biology is, to me, is something that, or rather, in my current role as in Learner Biopharma, is something that has not been proven to work in clinic because of constraints in drug technology, etc. But researchers have been working on and have all these new, fancy, cutting-edge, exciting ideas about how we can really treat diseases, or even recently, something that's very popular is to increase the health span of people. Yeah. Torsten, do you have anything to chime in?
Not much to add, no.
We discuss a lot.
Brilliant, thank you. So next sort of question sort of asks, in the translational space, a lot of academics use siRNA in their research. How can they begin to look to sort of turn it into therapeutics, I suppose? Not sure who would like to go.
I mean, I think I can go first. I mean, it's not anymore like so complicated in a sense that like if you have even like shRNA or siRNA, you make a screening. The biggest concern is like that the siRNA maybe which you use, or the shRNA, is specifically knocking down only in the animal model you're using, be it mouse, rat, or whatever. So like for each kind of like different species, you would have to design a new siRNA, make the modifications, and test again. There are some ways around. This is like this asset which Suhoi showed, like our prime asset. At Lerner, we have an algorithm which tries to find specifically sequences with efficient knockdown over the species barrier. So for this, we have efficient knockdown in mouse, rat, non-human primate, and human. So there is like one asset which can be going through the whole kind of like preclinical to clinical level. That said, this is not necessarily said that this is for all potential targets possible. So like this is one point where I think it's a concern like how to translate it. You have to test it again and again with different siRNA. Otherwise, it's nowadays quite straightforward to look for sequences. There are a lot of programs out there to test, and then you have to test for sure. It's not only like by bioinformatics. You have to test if you're on target and you have no off-target effects. This is like clear experimental biology which has to be applied.
Yeah, I will say that the generic siRNAs that is available to everybody through the mainstream vendors, they are really good tools for testing whether this mechanism of silencing the target works for your particular system, for validating the target, etc. But by creating a drug for that target is really a different ballgame from my experience. So as drug developers, we put a lot more emphasis about like what was the safety profile, the potency, etc. And we do so much screening. So with that, I hate to hate comparisons, and in general, once you go into drug development, you have to go fully in. So it's not really something that you can just take, a commercial sequence and say, okay, we got a drug.
Yeah, understandable. A few more questions coming in, and I say we have time for sort of 3 or 4 more. So the next one, asking about your LR1 sequence, any concerns with cross-reactivity between mice and human?
Like Torsten mentioned, when we developed the siRNA, we wanted it to be potent across multiple species. So of course, mice and humans are 2 of them, and therefore this sequence is cross-reactive against multiple species.
Okay. The next question, again, sort of any concerns about the ASGPR levels when working with a cirrhosis patient?
So we've looked into some of the human databases, and we find that the expression level of this GalNAc receptor ASGPR is actually comparable between normal hepatocytes from normal patients, normal livers, as well as cirrhotic livers. So we're not concerned.
Okay.
So maybe to add, is one thing is expression level. The important thing is for the receptor to be on the membrane, and there's sometimes there's some discussion about this. But we have stainings, or at least in NASH and this cirrhosis, that we see the receptor stained at the membrane. So we didn't went through all potential chronic liver modalities, but in this case, it's clearly there.
Right. Thank you both. I'd say sort of time for one more question. Just asking, with regards to the hepatocytes, obviously with the treatment, are there any concerns as to them growing too much after your siRNA treatments?
Torsten, your favorite question.
So this is how we tried. We also showed and we tried to mention. It's always a concern. So for example, even on the last Washington AASLD that was presented, one potential approach would be combination therapy of HGF and EGF, the major mitogens, and this mitogen combination can drive proliferation even beyond this kind of safety point where the liver normally stops, because it keeps a body weight to a liver weight ratio. What we're doing is in our system, we're checking if there's siRNA treatment induces proliferation without any damage. So we don't want any proliferation without an underlying damage. So it only should kick in and release the brake in these conditions. Second thing is for example, after partial hepatectomy, if we have the treatment, also the expression of siRNA, that we don't see an overgrowth of the liver. So that the liver regenerates, but the liver and body mass ratio is staying. Also, I mentioned short this FIH mouse. If you repopulate the mouse liver, we can see after repopulation, do we see still mitosis, or is the hepatocytes now quiescent, and that there's not an increase in liver mass? So it's a bunch of safety experiments which assure us that for this asset, we're on the safe side. But this, I have to agree, has to be evaluated for each asset individually.
Okay. Thank you, Torsten. I'm afraid that is all we have time for today. I'd like to thank our expert speakers, Dr. Torsten Fusterfeld and Dr. Si Hui Tan, for today's discussion presentation and this Q&A session. And a big thank you to everyone joining us online for submitting your questions. We hope that you found this a really worthwhile session. If you have any other questions, please feel free to email myself using the email on screen, editor@selectscience.net, and I will follow up with your questions after the webinar. If you'd like to listen to today's webinar again or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.
Thank you very much, Cameron.
Thank you. It's been a pleasure.